I was tempted to like bundle up before I came in here. All right, so we had an exam on Unit 1. So obviously now we're going to start Unit 2. So Unit 2 covers chapters 4, 5, and 6. The chapter 4 slides are already up on Canvas as of earlier this morning. I'll get the other 2 chapters up hopefully by the end of the week. Yeah, and so it kind of follows that same hierarchical set of steps we were talking about really early in chapter 1, right? So in chapters, starting in chapter 2 and through chapter 3, we talked about atoms, we talked about molecules. Chapter 3, we talked about macromolecules, right? What was the next step? Organelles. And so chapter 4, cell structure, AKA organelles. All right, and then chapter 5 gets into membranes specifically, right? That last little kind of slide of chapter 3 when we started talking about those phospholipid membranes. We're going to expand that out into kind of its own chapter in chapter 5, really pull that apart. And then chapter 10 is going to talk about cell division, so mitosis, basically. So it's a chunky unit, so brace yourself, prepare accordingly, study early, study often. There's a lot of words, there's a lot of stuff. So let's get in it. It's on the syllabus. I don't know off the top of my head. 3-ish weeks from now. They're kind of every 3 weeks, close to. So it should be something like that. Right, so cell theory, section 1. So we mentioned cell theory briefly and kind of the unifying themes of biology, right? We talked about kind of this unifying thing that all living things are made of cells. So we're going to talk about cell theory. We're going to talk a bit about why cells are small, because they are small. And then discuss kind of the major differences between different kinds of living cells. So lots of stuff about cells before we talk about the parts of cells. So cell theory, just to hit it one more time, right? It's got 3 pieces. All organisms are composed of 1 or more cells, right? And all life processes, right? Metabolism, heredity, yep, occur within them. Cells are the smallest living things, right? There's no organism smaller than or less than, rather, a single cell, right? And so it's kind of the organizing building block of all living systems, right? Everything is either a cell or it is made of a bunch of cells put together. And cells only come from the division of previously existing cells, right? Cells don't kind of arise on their own, right? It's that passing on of DNA, that inheriting of hereditary material, it's repetitive, right? That kind of makes new cells, right? You can't make a cell without using the instructions that are in the previous cell. Yes, so everything's made of 1 or more cells. The smallest cells are the smallest living things, and they're kind of the basic unit of all things, and cells only come from dividing already existing cells. So that is cell theory. Now, since everything's made of cells and cells are doing all the things, there are unifying features of cells. We'll walk through each of these in turn, but briefly, they are the possession of a nucleoid or nucleus, a place where all that hereditary information is kept. Cytoplasm, basically just kind of all the filler stuff that fills up a cell. What? It is like mucus. Ugh, not a mucus fan, but that's fine. If it works, it works. Ribosomes, right, which are the structures mostly made of RNA that make proteins, right? So they've got hereditary material, they're full of goop, they have things that make proteins, and they're all encased in a plasma membrane, right? That phospholipid, usually bilayer, right, that holds the whole thing together. So the nucleoid or nucleus is just your centrally located genetic material. This is like a cartoon of a bacterial cell, right? So in a prokaryote, right, that lack membrane-bound organelles, it's a nucleoid. So there's no membrane encasing the DNA. It's just kind of all balled up together in the center, right? And then in eukaryotes, right, like us, we have a nucleus. We'll talk about a nucleus later on, right? But it's just a membrane-bound organelle that holds the DNA, right? So even if you don't have a true nucleus, you still have all your DNA kind of bunched up together in some place. Right, so all cells have either a nucleoid or a nucleus. They all have cytoplasm, a semi-fluid matrix, mucousy even, jelly-like, right? And it fills the center of the cell, right? So everything has cytoplasm, the semi-fluid matrix made up of organelles, these structures that are specialized for particular functions, things like your mitochondria, your nucleus, your Golgi bodies and whatnot, right? So you're, what? Are we going to talk about it? Yes, not today, but we will. Wednesday, it'll happen Wednesday. Yeah. And then cytosol, which is all the kind of liquidy stuff that holds the ions and the molecules that all the organelles need to do all the things that organelles do, right? If they're building these macromolecules and doing metabolism, they need resources to do that, right? So your cytoplasm, this matrix of organelles and cytosol. Okay. Ribosomes, in the case of this photo, it's just all the tiny dots, right? And those are just structures made mostly of RNA, ribosomal RNA specifically, that synthesize proteins. So they're kind of found all over. They can be floating around on their own. They can be stuck to the membrane over here closer to the nucleus in the endoplasmic reticulum. We'll talk about it Wednesday, right? And so they are the structures that synthesize proteins. All right, so everything's got a nucleoid or nucleus. It's all got cytoplasm. All cells have ribosomes. And all cells are enclosed in a plasma membrane, right? Encloses a cell, all its contents. In the case of eukaryotes and bacteria, in this phospholipid bilayer, right? This is just a much more complicated version of what we saw on Wednesday, right? You've got these little non-polar tails towards the middle, these polar heads making up the outside of the layer, right? And when we get to chapter 5, we're going to pick apart all of these other things that are stuck to this plasma membrane. All right, but for right now, we'll keep moving. So cells have a place to put their DNA. They've got ribosomes, they've got cytoplasm, they've got plasma membrane, and they are generally small. And there are reasons, right? It's not just they just happen to be small. They're small because they can't be big. That sounds funny, but it's true. So cells rely on diffusion of substances, right? So things that the cell needs have to diffuse through the plasma membrane, right? And then move to wherever they're needed, right? And so that diffusion is affected by the surface area available, right? So the space that there is for things to come in at. Temperature, and that just falls back to that chapter 2 idea of like these reactions and the way that molecules interact. When things are warmer, right, molecules move faster, proteins are looser. If things are cold, things get really slow, right? So temperature plays a part. Concentration gradients of a substance, when we get into membranes, we'll pick that apart. But mostly, if there's a whole lot of something outside the cell that needs to go inside of the cell, it will diffuse faster because there's more of it there to diffuse, right? And then the distance over which the diffusion must occur. And this is where the cell size limitation really comes in, right? Because the cell is relying on things diffusing in and then getting where they need to be, right? And so as cell size increases, right, the distances in the cell are getting larger. And so as cell size increases, the time required to diffuse things from outside to wherever they need to get also increases, right? So the bigger the cell gets, the slower it would be at getting things where they need to be. Okay. So kind of compounding this, right? So my cell gets bigger. As things diffuse, it takes longer for them to get where they need to be. And because the cell is larger, it needs more macromolecules. It needs more energy. It's producing more waste. So by being bigger, It's moving things more slowly, but it needs more things. So it's kind of like doubly bad, right? Because it's taking longer to get what it needs, but it needs more, and then it's making more waste that then have to get out of the cell. So basically, if we're talking about evolutionary success, right, it is more efficient, more successful to have more smaller cells than fewer larger cells. So TLDR, cells are small, right? Because smaller cells have a larger surface to volume ratio, right? Because that rate of diffusion, the more surface area you have for things to come in, the faster you can diffuse, and the shorter the distance on the inside, right? The faster things will get where they need to be. And so if you are smaller, you have more surface area to unit volume versus something that is larger. Right? So as cell size increases, volume increases much more rapidly than the surface area, right? This is if you're in lab, those conversions where you were like enlarging 2-dimensional things versus 3-dimensional things, right? Your surface area enlarges as a 2-dimensional thing, while the volume is increasing, right, in this kind of threefold way. So volume gets larger faster than surface area. Right, and so small cells have more surface area per unit volume, and so they have more control over their contents. They can get things in and out faster, right? There are some exceptions. There are some bigger cells, but they kind of, not cheat, but they kind of make up for being larger, right? Neurons can be very long, but they're very, very skinny, right? So even though the cell is long in this direction, nothing coming in has to go very far, right? Because the cross section of the cell is very thin. Or things like skeletal muscles, which are also very large, actually have more than one nucleus to kind of mitigate the fact that it's got a longer distance to move things around, right? So cell size is limited. Subsequently, since cell size is limited, cells are small things, but if we want to observe them and talk about them, right, we need to see them. Obviously, you can't just look at individual cells. I can't just like see my cells if I look at my hand, right? Because most cells are less than 50 micrometers. And so you can't see them. This is due to limited resolution in your eye. This is just kind of a strange little tangent, right? But resolution is the minimum distance that two things can be apart and your eyes still know that they're two things. And the limitation comes from the size of the photoreceptors in your eye, right? So you're looking at stuff, right? And you see it because light is bouncing off of it, hitting your photoreceptors and telling your eye there's a thing there. For your brain to know that there are two things, the light that hits them has to hit two photoreceptors, right? If the light hits the same photoreceptor, your brain's just like, that's just one thing, right? Or it's all the same thing. So your photoreceptors, right? Things have to be at least 100 micrometers apart for the light to hit two different photoreceptors. Anything smaller than that, your brain can't see as two things. So we use other tools. That's fine. We use microscopes. In the next week or two, I don't know when, is your lab practical this week? You're not in the lab. Next week. I'm not sure where I'm not teaching 1101 labs. I don't know where they're at. I don't think so. They don't really track much after that. There's just a couple of things where so like the 1101 lab doesn't track with 1101 class. There are just a couple of things we talk about that are the same. Because the 1101 lab, because it's got that gen ed component, actually the whole last 3 labs are part of the gen ed project, and it's complete own thing. But the microscopy and unit conversion are just something that we talk about in both of them. Yeah. So if you've got your practical, I think microscopes happen after that, but you'll be using light microscopes, which use visible light and a couple magnifying lenses to make an image where the things you're looking at appear bigger because you're using magnifying lenses. You can have compound light microscopes. I think this is technically what we have in the lab, where you've got several lenses kind of bouncing things around and enlarging images. And so you can see things that are separated by at least 200 nanometers. So, right, orders of magnitude even smaller than what we were just talking about. But cell membranes are only 5 nanometers thick, which means that even with a compound microscope, we can't really see the parts of a cell membrane. So we've got other kinds of microscopes. We'll talk about just a couple of them. Really one and a couple flavors of the one. So there are electron microscopes, which is cool, right? Because the problem with the light microscope is when you're looking at things that are sufficiently small, you are actually limited by the size of a wavelength of light, right? So to see things smaller than the width of a wavelength of light, you've got to find something smaller than light. Wow. That would be electrons. So there are microscopes that use beams of electrons, right? Because they can resolve even smaller things. They have a higher resolution. Basically, there's two main types, transmitting and scanning electron microscopes. So transmission electron microscopes, TEMs, shoot electrons through something. And you get these kind of two-dimensional, kind of black and white images. They use these a lot in histology, like if you're doing histological sections at a hospital or in a lab here, right? So this is a cross-section of some tissue that's been like flash frozen. You pass electrons through it and you can get images of all the structures in the tissue, right? And can resolve objects that are 0.2 nanometers apart, which is really, really freaking small. I've never used a TEM, have I? I don't think so. I've used SEMs. SEMs, scanning electron microscopes, beam electrons at an object and then records the electrons bouncing back. So that's kind of how it takes in information. The electrons are reflected back and you get a 3D image, which is kind of fun. And you get this like with these lit up areas because when you use a scanning electron microscope, actually, if you ever have an opportunity to, it's super fun. So you get these little, they're called stubs, but it looks like a blunt thumbtack almost. And you put whatever you want to look at on top of the stub, and you put that in this crazy chamber. Well, you hit a button, whatever, and it sputter coats it with gold, right? So that it will reflect electrons. So you take that out, you put it in the SEM, which is this big machine, like the size of this podium or larger, and then you get these images, right? You get these three-dimensional images. Sometimes people will color them. They're very cute. If you're into microscopy, I kind of like microscopy. I think it's fun. But yeah, so if we need to see things smaller than we can with light microscopes, we can use electron microscopes, right? We can add stains to things. Again, if you're in the lab, I think you'll have a mix of stained and unstained things, right? Because I can put a piece of a leaf on a slide and just look at it, and it's mostly just going to kind of look like this. It's going to be hard to see things, hard to tell things apart. You can get stains where you can see things like cell walls. You can see nuclei, right, kind of popping out. Nuclei are really big, so they're usually pretty easy to see, right? And so we can use stains to kind of help us visualize stuff. All right. This is just kind of a breakdown of what you need to see different sizes of things. We'll let it go. Right, so cells are small by necessity. Step next, we're going to kind of break cells out into two main groups, right? So we're going to talk about prokaryotic cells for a little bit. This is the bacteria and archaea. And then we'll start talking about eukaryotes, and we'll spend the majority of the rest of our time this semester, honestly, with eukaryotes. Right, so for prokaryotes, we're going to talk about basic structure. They're pretty simple. And then talk a bit about bacteria versus archaea. So prokaryotes are the simplest organisms, right? Definitively, prokaryotic cells lack membrane-bound organelles. So they do not have a true nucleus, right? They've got that nucleoid instead, this kind of all their DNA all kind of tucked together. And that DNA in the nucleoid is usually circular, right? So instead of, we have kind of long strands of linear DNA for the most part, prokaryotes tend to have DNA that actually forms a circle. They have little rings of DNA. And they do contain ribosomes, because as we'll learn in a little bit, ribosomes are not membrane-bound structures. There's something a little different, right? So we're not breaking any rules by having ribosomes. So we've got our nucleoid, got our ribosomes, and then a few other things that are more particularly bacterial or prokaryotic, right? So we still have a plasma membrane, right? Everybody's got a plasma membrane surrounding their cytoplasm, right? Ribosomes, nucleoid. But outside of that, bacteria in particular will frequently have a cell wall, different from a plant cell wall, and occasionally a capsule, which is kind of this thick, sticky layer that will be on the outside of some bacteria. And then they may have some extra structures on the outside, these things called pili, which are just kind of little extensions of the plasma membrane. They look kind of like little hairs. And then they might have a flagellum. It's a bit different from the flagellum we see in eukaryotes, but the same thing, kind of used for locomotion or moving around. Right, so we've got this general prokaryote structure, and this is kind of all they've got going on in the broad scheme of things. All right, so there's not a lot there. So they're simple. There's not a whole lot to them morphologically, but they are really, really important in living systems, in just human systems in general, right? They can be photosynthetic, right? Which is always kind of important. They break down dead material. They're really important in decomposition of dead and dying stuff. They also are obviously, right, medically important. We have pathogens, bacteria, and that will cause disease, so they become medically important. And then they've got industrial or kind of economic importance, right? Kombucha, beer, bread, cheese, wine, anything that's made with any kind of fermentation process is done using bacteria. And so kind of whether you realize it or not, you are interacting with bacteria literally all the time. I mean, it's on everything to start. It's in you, right? You're also kind of full of bacteria. I don't have the statistic off the top of my head, but there's an absurd number of the cells in your body that are bacteria and not you. Look that up. It's a weird number. Well, not weird. Alarming, maybe? I don't know. But right, so they're important. They're very diverse. Now, because they're so kind of morphologically simple and similar, classifying them can be a bit difficult, but it's estimated that there's between 1 and 100 million species, right, of prokaryotes on Earth. Right, and so people are working on that. We split them into 2 domains, right? And so we divide them into the bacteria, which we hear a lot about, and the archaea, which we hear less about. But we'll kind of break that down just a smidge. So differentiating bacteria and archaea. Bacteria have a cell wall, and it's, like I said, it's different than a plant cell wall, right? Plant cell wall is made of cellulose. Bacteria cell walls are made of this peptidoglycan. So it's carbohydrates cross-linked with short polypeptides, right? So it's got peptides, there's your peptido, it's got some kind of carbohydrates, that's your glycan, right? So they have the cell wall made of peptidoglycan, right? Which animal cells don't have. Your cells don't have this, which is really handy because occasionally bacteria get in your body that you don't want to be there, right? And so this is the way some antibiotics work, by targeting either that peptidoglycan cell wall or bacteria's ability to make that cell wall. Subsequently, they can kill the bacteria and you can cure whatever is ailing you at the time, right? So penicillin and vancomycin work that way. They interfere with bacteria being able to specifically cross-link those polypeptides, but basically make them incapable of making a cell wall. The bacterial cells break down and you get whatever nasty bacterial bug you've gotten out of your system. Some bacteria secrete that, like that thick red layer that was on that picture, a jelly-like capsule. We call it a capsule, and it makes them sticky, right? And so you see this in a lot of disease-causing bacteria because it allows them to stick to things they're trying to infect, basically, right? To surfaces like your teeth, your skin, your food, right? Those kind of biofilm sort of situations that bacteria will create, right? And so you see this kind of sticky outer layer on a lot of disease-causing bacteria. All right, So that's bacteria, as far as we're really going to cover them in here, versus archaea, right? And so these are also prokaryotes, but they differ from bacteria in a few ways. They generally lack cell walls. So they lack, generally speaking, they don't have that peptidoglycan layer. And they frequently have a monolayer plasma membrane, which kind of breaks all our rules, right? Because everything's got this lipid bilayer. There are some archaea that only have a monolayer plasma membrane, which somehow, and this is just my lack of knowledge of physics and chemistry, makes them more thermally stable. You tend to find archaea in a lot of extreme environments at like thermal vents and stuff, right? But they also kind of interact with us. They're not known to be parasites or cause disease, but they are found in your gut. You do have gut archaea that aid in digestion, so they're important in that sense. They're used in biogas production and sewage treatment. There's a new, and that's actually technically what these are supposed to be pictures of. Asgard archaea were discovered a few years ago, and they're interesting because when scientists started looking at the DNA of the Asgard archaea, right, where you had, so this kind of drew like the relationships of all stuff, right? You've got bacteria, archaea, and then eukarya. When you add in the Asgard archaea, eukarya becomes nested inside the archaea, which technically, potentially, could mean that we are all just highly evolved archaea, which is a weird thing to think about, but it's possible. It's kind of a new idea coming through in science currently. All right, so that's archaea. Don't have cell walls, generally speaking. Sometimes weirdly have a monolayer plasma membrane. Do not cause disease. All right, that's enough prokaryotes. Eukaryotes. See if we can get through. This will be the last section we cover. We'll see how far we get. So we're going to compare briefly eukaryotic and prokaryotic cells, given what we know now about prokaryotes. And then we're going to start kind of working through the structures in a cell, starting at the nucleus and kind of working our way outward. It's going to be the general process, right? And we'll end this section at ribosomes and protein synthesis. So, eukaryotic cells, unlike prokaryotic cells, possess an endomembrane system, right? We've got a plasma membrane like everybody else, but we also have this internal membrane system that divides the cell into compartments, right? This is what prokaryotes don't have this, right? So they don't get this kind of diversity of structures, organelles, right? So your cell has this kind of folded up membrane system inside it, right? That holds your nucleus, right? Holds your Golgi body, endoplasmic reticulum, all these little bits and pieces, and that is supported inside the cell by the cytoskeleton, which is kind of this internal made of protein, this scaffold that basically holds all the pieces in your cell where they're supposed to go, right? And so this kind of level of complexity is kind of what differentiates us from prokaryotes. Ugh, there we go. Right, so this endomembrane system creating these organelles that are held in place by the cytoskeleton. So starting in the center, nucleus. It's the largest organelle if you start looking at cells, right? Like that kind of slide picture we had before. Nucleus is the giant big circle in the middle. Holds the genetic information for synthesis of most of the proteins in the cell, right? All your DNA, or all of your linear DNA is held in the nucleus, right? Stored as multiple linear chromosomes. In our case, 2 pairs each of 23 different chromosomes, right? And that whole noodly mess is organized, right? Kind of like you would wind up string on a spool. Your DNA is organized with proteins into this kind of spooled up structure called chromatin, right? So it's not just kind of all loosey-goosey spaghetti mess in the middle. It's all kind of tidied up, spun around these proteins so that it stays nice and organized, okay? So that's what's going on in the nucleus. In the center of the nucleus, there's this kind of, they always just kind of draw another darker circle in the middle. That's the nucleolus. In the center of the nucleus, and this structure is specifically, right, because all of your DNA, when you express things, genes and whatever, right, pieces of RNA are being copied from your DNA. But the nucleolus specifically is for synthesizing ribosomal RNA. Right? So it's making the rRNA that will become your ribosomes, and that is its only job. Right there. So you've got a nucleus, your biggest organelle in the center is your nucleolus, which is just in there cranking out rRNA. And all of this is encased in a nuclear envelope, right? It's kind of what this orange is supposed to be denoting here. So the nuclear envelope, kind of the membrane system around your nucleus, is actually made of 2 phospholipid bilayers. So you've got this inner membrane, and it's just on its own, just the membrane around your nucleus. But there's a second layer, right? And the second layer around the outside is actually continuous with the rest of your endomembrane system, specifically the endoplasmic reticulum, right? Which is always just kind of stuck right here to the outside of the nucleus. And it's because they share this outer membrane, okay? So you've got your nucleolus, nucleus, its little individual membrane, and a second membrane that ties it to the ER. All right, now, my nucleus is wrapped in 2 membranes, but there's little pores, right? So they're kind of little pinched in areas where the 2 membranes meet, right? Which allows ions and molecules as are needed to pass in and out of the nucleus, right? Because if we're synthesizing RNA in here, we're going to need ions and small molecules, and then we're going to want to pass those back out. So we've got these pores in the membrane where things can get in and out of the nucleus through that double membrane. All right, so nucleolus, nucleus, double membrane with pores. Cool. All right. And just again, we mentioned this already, but we'll mention it again, just kind of along with the nucleus. Inside that nucleus is all your DNA, all these linear chromosomes, but it's all spooled up around these histones, or what these little proteins are. That's fine. But basically, it's wound around these little proteins, coiled up and stored until it's needed. And then whatever little piece is needed kind of unwinds and then winds back up. All right. So that nucleolus is cranking out this rRNA, this ribosomal RNA. And this is where, right, prokaryotes have ribosomes, which look in pictures weirdly like organelles, but they're not organelles. They're actually just 2 strands of rRNA, right? They're just like a huge, huge molecule, right? And that is your ribosome. So your nucleolus is transcribing or synthesizing rRNA to make the large and small subunit of a ribosome, right? There's a couple proteins stuck in there too, but mostly it's just rRNA, right? Use a little cartoonier picture, right? So you've got like a large subunit and a small subunit, right? They only come together if they're synthesizing a protein. If you remember the rRNA or the RNA slide, right? We had rRNA, it's what the ribosome is made of. We have mRNA, which is RNA that's carrying some message to make a protein. And so when an mRNA shows up, one of these small and large subunits kind of clamps down on it, right? And then it starts reading this messenger, the message from the messenger RNA, to produce an amino acid chain. Okay. What level of protein structure is that amino acid chain then? Just the chain. String of amino acids. It's definitely on that exam on Friday. What level of P? What level of structure? Something structure. That starts with a P. What? Primary structure. Right? It's level 1, right? We've just made we've made the string of the protein, primary structure produced by the ribosome. Right? So, messenger RNA is carrying some information for making a protein. rRNA clamps on it, tRNA is the other RNA. And so it's bringing the amino acids in. So it's like the little kind of runner guy, right? So you've got mRNA. This is the protein we need to make. mRNA, rRNA is making the protein. tRNA is bringing the amino acids, right? And so these ribosomes making these proteins are found in the cytoplasm, kind of all over, right? You remember that picture of the cell? They were just kind of like little dots all over the place. So they can be out in the cytoplasm or attached to the membranes in the endomembrane system. So attached to the outside of organelles, right? They can kind of be wherever it is they need to be making proteins. All right, which gets us to the endomembrane system, which is a fantastic place to stop because we're basically going to start with that mRNA and those ribosomes and walk all the way through the process that happens making proteins and other things in the cell on Wednesday. So ribosomes is a good place to pause. All right, so with that, I will see you on Wednesday.